Chapter 1. The cell as a unit of health and disease. These are the objectives. Introduction to pathology. Adaptation of cellular growth and adaptation. Reversible cell injury. Irreversible cell injury. Intracellular accumulation. Cellular aging. Introduction to pathology. Logos means, study, and pathos means, diseases. Pathology is the study of structural, biochemical and functional changes that underlies disease. It uses molecular, microbiologic, immunologic, and morphologic techniques. Adaptation of cellular growth and adaptation to environmental stress. Hypertrophy is an increase in the size of an organ or tissue due to an increase in the size of cells. Other characteristics include an increase in protein synthesis and an increase in the size or number of intracellular organelles. A cellular adaptation to increased workload results in hypertrophy, as exemplified by the increase in skeletal muscle mass associated with exercise and the enlargement of the left ventricle in hypertensive heart disease. Hyperplasia is an increase in the size of an organ or tissue caused by an increase in the number of cells. It is exemplified by glandular proliferation in the breast during pregnancy. In some cases, hyperplasia occurs together with hypertrophy. During pregnancy, uterine enlargement is caused by both hypertrophy and hyperplasia of the smooth muscle cells in the uterus. Aplasia is a failure of cell production. During fetal development, aplasia results in agenesis, or absence of an organ due to failure of production. Later in life, it can be caused by permanent loss of precursor cells and proliferative tissues, such as the bone marrow. Hypoplasia is a decrease in cell production that is less extreme than in aplasia. It is seen in the partial lack of growth and maturation of gonadal structures in Turner syndrome and Klinefelter syndrome. Atrophy is a decrease in the size of an organ or tissue and results from a decrease in the mass of pre-existing cells. Most often, causal factors are disuse, nutritional or oxygen deprivation, diminished endocrine stimulation, aging, and denervation, lack of nerve stimulation in peripheral muscles caused by injury to motor nerves. Characteristic features often include the presence of autophagic granules. Metaplasia is the replacement of one differentiated tissue by another. There are three types of metaplasia: squamous metaplasia, osseous metaplasia, and myeloid metaplasia. Squamous metaplasia is exemplified by the replacement of columnar epithelium at the squamocolumnar junction of the cervix by squamous epithelium. It can also occur in the respiratory epithelium of the bronchus, in the endometrium, and in the pancreatic ducts. Associated conditions include chronic irritation, squamous metaplasia of the bronchi with long-term use of tobacco, and vitamin A deficiency. Osseous metaplasia is the formation of new bone at sites of tissue injury. Cartilaginous metaplasia may also occur. Myeloid metaplasia, extramedullary hematopoiesis, is proliferation of hematopoietic tissue at sites other than the bone marrow, such as the liver and spleen. Carbon monoxide poisoning, which results in diminution in the oxygen carrying capacity of red blood cells by chemical alteration of hemoglobin. Decreased perfusion of tissues by oxygen carrying blood, which occurs in cardiac failure, hypotension, and shock. Poor oxygenation of blood secondary to pulmonary disease. Early stage hypoxic cell injury first affects the mitochondria, with resultant decreased oxidative phosphorylation and adenosine triphosphate, ATP, synthesis. Consequences of decreased ATP availability include failure of the cell membrane pump, Wabba insensitive sodium potassium ATPase, results in increased intracellular sodium and water and decreased intracellular potassium. This process causes cellular swelling and swelling of organelles. Cellular swelling/hydropic change is characterized by the presence of large vacuoles in the cytoplasm. Swelling of the endoplasmic reticulum is one of the first ultrastructure changes evident in reversible injury. Swelling of the mitochondria progresses from reversible, low-amplitude swelling to irreversible, high-amplitude swelling, W. Swelling of the mitochondria progresses from reversible, low-amplitude swelling to irreversible, high-amplitude swelling, which is characterized by marked dilation of the inner mitochondrial space. Disaggregation of ribosomes leads to failure of protein synthesis. Ribosomal disaggregation is also promoted by membrane damage. Stimulation of phosphofructokinase activity results in increased glycolysis, accumulation of lactate and decreased intracellular pH. Acidification causes reversible clumping of nuclear chromatin. Late stage. Hypoxic cell injury eventually results in membrane damage to plasma and to lysosomal and other organelle membranes, with loss of membrane phospholipids. Reversible morphologic signs of damage include the formation of myelin figures, which is a whorl-like structures, probably originating from damaged membranes, cell blebs, a cell surface deformity, most likely caused by disorderly function of the cell death. Finally, cell death is caused by severe or prolonged injury. The point of no return is marked by irreversible damage to cell membranes, leading to massive calcium influx, extensive calcification of the mitochondria, and cell death. Lactate dehydrogenase LDH. Creatine kinase, CK, also known as CPK. Aspartate aminotransferase, AST, previously known as serum glutamic oxalic acetic transaminase, has been used in the past but has fallen out of favor due to poor sensitivity for myocardial infarction. These markers of myocardial necrosis vary specificity for heart damage, as well as in the time period after the necrotic event in which elevations in the serum appear and persist. The delineation of isoenzyme forms of LDH and CK has been a useful adjunct in adding specificity to these measures. The foregoing enzymes are beginning to be replaced by other myocardial proteins in serum as indicators of myocardial necrosis. Important examples include the troponins, troponin I and troponin T, and myoglobin. Liver enzymes in serum. Enzymes of special interest include the transaminases, AST and alanine aminotransferase, alkaline phosphatase, and gamma-glutamyltransferase. The vulnerability of cells to hypoxic injury varies with the tissue or cell type. Hypoxic injury becomes irreversible after 3 to 5 minutes for neurons. Purkinje cells. The cerebellum neurons of the hippocampus are more susceptible to hypoxic injury than are other neurons. 1 to 2 hours for myocardial cells and hepatocytes. Many hours for skeletal muscle cells.